Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, honorable examiners. Let me introduce myself. My name is Eva Rosita with student ID number 12320600077, and I'm from Biology Education Study Program. Today, I would like to present my research proposal titled The Influence of Using Smartphone Assembled Microscopes on Student Science Process Skills in Cells Biology and Materials. I would like to express my gratitude to my academic supervisor, Ms. Maryanti M.Pd, and my course instructor, Mrs. Asrianty, S.Si M.Pd. With your permission, I will begin the presentation. There are three main problems behind this research. The first problem is students are not yet trained in their SPS or science process skills, such as observing, classifying, interpreting, communicating, and applying concepts. As a result, they face difficulties during the learning process. Second problem is cell material is abstract. Cell topics are microscopic. It is difficult for students to imagine the cell structure if they only learn from static like picture or videos. The third problem is microscope limitation. The number of microscopes at the school is limited. Many are broken or moldy. Therefore, science learning is often replaced by just using picture or videos. Students lose hands-on learning experience. The solution I offer is using smartphone assembled microscope. It is cheap and the material are easy to find, and it can be self-made. We not use digital microscope because they are expensive and require electricity. This research is supported by several data showing that smartphone microscopes are effective for science education. And Pratiwa Engineering said that showing that SPS increases their cell practice work. The novelty of this research is using an assembled microscope that is cheap and easy to make, which is different from previous research. Additionally, the study has never been conducted at SMAN 26 Bandung. Based on the background, I have formulated four research questions. First is, how is the implementation of learning using smartphone assess assembled microscope in cell biology materials? Second, what is the difference in science process skills between students who use smartphone assess assembled microscope and students who do not use microscopes in cell biology materials? Third, what is the influence of using smartphone assess assembled microscope on students' science process skills in cell biology materials? Fourth, how do students respond to the use of smartphone assess assembled microscope in cell biology materials? On this question, the research objectives are: first, to describe the implementation of learning using smartphone assess assembled microscope in cell biology materials. Second, to analyze the difference in science process skills between students who use smartphone assess assembled microscope. Third, to identify the extent of the influence of using smartphone assess assembled microscope on students' science process skills in cell biology materials. Number 4, to analyze student response to the use of smartphone assess assembled microscope in cell biology material. The study provides two main benefits: theoretical benefit and practical benefits. The theoretical benefits: this research is expected to contribute to the development of educational science, especially regarding simple technology in biology education. And for practical benefits, for students, it improves their science process skills. For teacher, it serves as an innovative and affordable learning alternative. For school, it provides a solution for conventional microscope limitation. And for researchers, it broadens insight into technology-based media. For previous research, I included 3 research out of the 10 research that are in my proposal. So from this 3 research, it is said that the use of media such as augmented microscope can improve students' science process skills. And for the research gap, there is no research that specifically examines the influence of using smartphone assist assembled microscope on students' science process skills in cell biology materials. Now I will explain the learning framework. This framework starts with analyzing the learning outcomes. In phase F, students must have the ability to describe cells. From these learning outcomes, or CP, I developed 6 learning objectives, or TP, and 6 achievement indicators, or IKTP. These indicators are aligned with 6 science process skills, or SPS indicator, berutama: observation, classifying, identifying, communicating, measuring, predicting, and concluding. In this study, I will compare two classes, the experimental class and the control class. The experimental class will use smartphone assist assemble microscopes. Students will do direct observation, classify cells, interpret function, communicate results, measure cell scales, and predict structure. Control class will not use microscope. Learning is done through picture, videos, and or textbook, covering introduction, material delivery, Q&A, and practice exercise. The strength of the experimental class are direct observation, training all 6 SPS indicators, and providing meaningful experience. The weaknesses are that it requires tool preparation and intensive supervision. For the control class, the strengths are that no tools are needed, making it easy and efficient. However, the weakness is that students lose hands-on experience, so their SPS is not trained. The expected result is that the science process skill of student is the experimental class will be higher than more optimal compared to the control class. So this is our research hypothesis for my proposal. Based on Piaget's constructivism theory, direct observation and exploration can increase students' skills. Therefore, the theoretical hypothesis states that smartphone assist assemble microscope enable students to learn more actively and independently. The research hypothesis is the use of smartphone assist assemble microscope has a significant influence on improving students' science process skills in cell biology materials. Statistically, this means age 0 is rejected and age 1 is accepted. We're moving on to the methodology, approach, and method. This study uses a quantitative approach with quasi-experimental design. This design is non-equivalent control group design, meaning the two classes are not selected randomly because they were already established by the school. Population and sample. The population is grade 11 science student at SMA N26 Bandung. The sample consists of two classes with 30 students in experimental class and 30 students in the control class, making a total of 60 students. And for the samplings, I use purposive samplings because of benefit criteria, such as both classes having relatively well academic capabilities and having never used assembled microscope before. I will use 3 research instruments. First is test. I will give 10 essay questions for pre-test and post-test to measure SPS. Next is observation sheets to assess 6 SPS indicators during the practical work, and questionnaire. There are 10 statement Likert scale to measure students' response given only to this experimental class. Before use, all instruments will be validated through experiment judgment. The data collection technique is straightforward. I will give a pretest at the beginning, then the experimental class will do practical work using the assembled microscope, while the control class learn from picture or videos. At the end, a post-test will be begun and the questionnaire will be distributed to the experimental class. The research procedure consists of 3 stages. The first is preparation stage, implementation stage, and final stage. The preparation stage is conducting preliminary studies, literature review, creating instruments, validation, instrument testing, obtaining research permits, and building the assembled microscope. And for the implementation stage, administering the pre-test, conducting the learning treatment for 1 meeting, administering the post-test, and giving the response questionnaire. And for the final stage, I will processing the data, conducting data analysis, drawing conclusion, and writing the final report. Lastly, for data analysis techniques, I will explain them based on each research question. For the first research question is for learning implementation. Data from observation sets will be analyzing using percentage and categories from very good to very poor. For the second research question, for the difference in SPS, data from the pre-test and post-test will be analyzed using the independent sample t-test if the data is normal and homogeneous. If the data is not normal, the Mann-Whitney test will be used as the alternative. For the third research question is influence on SPS. I will calculate to the end gain to see the improvement and cohence the effect size to identify the magnitude of the influence. And for the last research question is for student response. Data from the Likert scale questionnaire will be analyzed using percentage and category from very positive to very negative. Thank you very much for your time and attention. I look forward to receiving your valuable feedbacks, questions, and suggestions to improve this research. Wassalamu'alaikum warahmatullahi wabarakatuh.